Well, welcome everyone. I'm Arthur Gosnell, and it's my great pleasure to introduce our speaker this evening, Ken Nedimyer. Ken is from the Florida Keys, and he is considered to be one of the leading experts across the globe on reef restoration. In 2007, Ken established the Coral Restoration Foundation, originally a bootstrap startup. The CRF last year had an annual operating budget in excess of $3 million. Through his work as the founder and the longtime president of the CRF, Ken has been instrumental in restoring extensive areas of degraded reefs along the Florida Keys, as well as here in the West Indies and as far away as Australia. Excuse me. It's difficult for me to quantify the extent to which Ken has had an impact, but it's fair to say that he and the teams he has supervised have grown tens of thousands of baby corals. More recently, Ken has founded the Reef Renewal Foundation International, and he is the technical director of Reef Renewal USA. So he is at it again. We've been extremely fortunate to have Ken come to Antigua this past week, where he worked extensively with Molly Wilson and Genevieve Renaud Byrne, our science team from the Elkhorn Marine Conservancy. And it's great to have both Molly and Jen with us this evening. So Ken has done it all, and in recognition of this, he was named a CNN hero in 2012, a Disney Conservation Hero in 2014, Scuba Diving Magazine's Sea Hero of the Year in 2014, and received the Whalen Icon Award in 2013. And on top of all that, he is a super nice guy. So please welcome Ken.
I think it's working. Wow. Echo. The sound guy is on top of it. So thank you very much. This has been a long time coming to be here. Everybody probably knows Martha. Where is Martha? Yeah, so this is really Martha's fault and Martha's dream. I met Martha in probably 2015 at one of our fundraisers in the Florida Keys, and she generously donated not just 1, but 2 weeks at her treetop resort. What do you call it? Cottage. And we ended up raising money and unrestricted funds. And anybody in the nonprofit world knows that unrestricted funds are gold. And so I thank you. So I'm going to try to get started here. Let's see. Yep. Okay. So tonight I'm going to just try to talk about who I am, why I'm here, and some of you know that. I'm not going to go into great detail on who I am, but I'm going to try to tell you why I'm here. A lot of it is for a site survey, so we'll talk about the site survey results, and then we'll talk about some recommendations that we might have, and I'll just show you some of the results we've had elsewhere. And it's going to be light, a lot of pictures, no graphs, no charts, some text slides, not many, but a lot of pictures, and hopefully you'll enjoy it. I would normally say raise your hand if you have a question, but I think we'll run out of time. There's going to be a question and answer period afterwards, and we can try to go through it then. And I can always go back on slides if you want. And so who am I? I'm Ken Nedimyer. I've grown up in Florida for almost all of my life. My dad was in the space program. We put a man on the moon when my dad was working there, and it was exciting to see it happen. But as soon as I could, I got out of college and I moved to the Florida Keys because my heart and soul was in the water, not in space. It was coral reefs or bust. And so I moved to the Florida Keys in 1978 after getting out of college. I tried to get a job in marine biology, which is what I was trained to do, and there was just no jobs available. It was like, you had to have a PhD, the pay wasn't that good. So I turned to commercial fishing, which seems weird, but that's what I did for a long time. And it was a weird type of commercial fishing. It was tropical fish collecting. So a lot of people would never even look at that as commercial fishing. But it was. I made a good living. I was really good at it because I think my science background helped me understand tides and larval distribution of fish and yada, yada, yada. But I got really good at it, and I made pretty good money at it, but I was watching the reefs dying around me. And as the reefs died, as the corals died, the fish weren't coming back. The fish don't have to have coral, but they really prefer coral. Matter of fact, larval fish can smell a coral reef from a long distance off and are drawn to it. They smell it and they hear it. And so they're drawn to it. So when there's live coral, there tends to be more fish. And I was seeing the evidence right away. And I thought, I've got to do something. What can I do about that? And so it's a long story, and I'm not going to go into that. But in the mid or early 2000s, I stumbled onto I changed my style. I went from being a tropical fish collector to a coral farmer. And initially it was a coral farmer to make money. I was going to sell these corals, long story there, but I was going to sell the corals and make money in the tropical fish industry. I never sold the first coral because I started getting this idea, what if we could put them out on the reef and restore these reefs? And so I got permission to put some corals out on the reef, and I started watching them. I was just like, this is what I want to do. So I started doing that, and that was 2001. In 2007, I formed this Coral Restoration Foundation because I realized I needed a vehicle to advance this thing forward. I couldn't get funding, I couldn't get grants being Ken in a boat, and I needed to have a mechanism to do it. So I did that. We went from a zero budget to, as Arthur said, last year it was over $3 million budget. It's the biggest coral conservation, coral restoration organization in the world. I created the biggest coral nursery in the world when working there. And, we really stepped the game up, and everybody saw our successes. And it really, from 2001 to now, we've gone from people not believing this could happen, and not really knowing what we were doing, to a huge organization in the Keys and a huge impetus in restoring coral reefs in Florida. I think there's what they're calling the Iconic Reefs project in Florida now. We've picked 7 different reefs, and we're really going to hit them hard. And it's $100 million over the next 10 years is what they expect it to cost. And they're looking to raise $10 million this year, and I think they're more than halfway there. So we went from no money in conservation, no money doing that, to $10 million this year. So it's really exciting. I need to move on. Anyway, so during this time when I was at the Coral Restoration Foundation, we would start having these annual galas where we would try to raise these unrestricted funds that I mentioned earlier that are like gold, because you can do a lot of things. You get a lot of grants that won't fund overhead, they won't fund this, they won't fund R&D, they'll only fund a certain thing. So when you can get unrestricted funds, then you can fund the things that you need for overhead, and you can also fund some research. And so I met Martha there. She generously donated weeks at her treetop guest cottage, and she started saying, could you come to the Antigua and do it down here? And I said, well, sure, I'd love to. And it wasn't that easy. And I kept telling Martha, I said, Martha, you need to find somebody in Antigua that's going to take ownership of this project, because you're not going to do it. You need to find somebody that'll do it. And because my goal all along was to help people do this, but not own the project. I want the local ownership to take over, and I'm just going to help you. And I just kept telling Martha, you got to find somebody. And about a year ago, I got an email from, I think, Jen or Molly, one of the two of them, and they started, so Martha had found somebody. And I can't express enough praise for these two girls. They are really sharp. They put together a really good baseline assessment of the reefs out here. I read through it. I was like, wow, these girls are great. And you're really lucky to have somebody that wants to do this, and it's really professional. A lot of times we go to different islands and we set up a program. We're dealing with a dive master who's never gone to college, who can't use a computer, but who wants to do it. So now we have somebody that's gone to college, has a PhD or a master's degree, knows how to use a computer and spreadsheets, and they're excited about it. So you're really lucky, and I'm really excited about having them aboard. I need to move on. So why am I here? I was here to do a site assessment. So Molly and Jen have already done that, but we needed to get it. I wanted to get, if they want to get involved in this and they want to involve me and our organization, I said, look, it'd really be best if I could see what you've done and get my eyeballs on the reef and just verify that we're on the right track. So that's why I'm here, and that's what we've been doing for the last several days. So I'm going to move on. Click. So this is just for this international organization that I'm a part of. I founded it, but I don't want to be the owner of it. I wanted to have business people running it, and I'm like the guy that goes out and does these things. And so there's a role that I play, and I do really well, but I don't run businesses really well. I don't like to write checks. I don't like to do that stuff. So I have a group of people that do that, and I do what I like to do and I'm good at. So these are just some of the places in the Caribbean where we've been working. I've also set up nurseries in Moorea in French Polynesia and Australia. And there's a lot of other places that have wanted me to come, but I said, look, I only have a certain amount of time, and I can only go so many places. So that's the way it is. And I haven't gone anywhere in the last year. So this is the first trip I've taken. I'm excited. So a lot of the work and a lot of what we're going to be talking about tonight are these two corals here, Staghorn Coral and Elkhorn Coral. And Elkhorn Coral in particular, the one on the right, it was the dominant coral here in Antigua and throughout the Caribbean. It was, by and large, the most common coral in the reef. And we've gone out here all over, and everywhere you go, you see remnants of that particular coral. So it's a super important coral. And it's also really easy to grow. It's easy to propagate. It's easy to work with. And so that's where we're going to start with this whole project. These two corals, and there's actually a hybrid that's in between them. And the hybrid can, I was telling somebody last night, it's like if you have Elkhorn Coral over here and Staghorn Coral over here, and they spawn and they mix up, there's something in the middle. But then that one in the middle can spawn with the one over here and become something like this, or it can go this way. So there's a whole suite of hybrids in the middle of the Elkhorn and the Staghorn Coral. And so, but we're going to work with a couple of those because they're really hardy species, and there's a lot of them here. And why not? They're easy. So if you had, now I should say also, all of these images were taken in the last 10 years. So these kind of scenes still exist today, but they used to exist everywhere. If you would dove out here in certain places, certain habitats in the 70s, this is what it would look like. You'd have all these boulder corals, Staghorn coral filling in all the gaps, just thickets of coral, fish down in there, lots of habitat, lots of places for fish to hide. This Elkhorn coral formed all these reefs out here. And when you go out there and anywhere from 5 feet to 25 feet, there's just field after field of the skeletons of this. This is what it looks like now, just all dead. And most of it dead, 95% of it. 95% of it died in the 1980s, and probably 2% has died since then. So it's bleak, it's not pretty, and it's depressing. And when I see all these reefs, I want to cry because I remember what they used to look like. And so I think that's what's driven me, is I know what they used to look like, and I think there's still hope. And I'll talk about the hope. I don't want to depress you. It is a depressing story. So Staghorn coral is the other coral I was talking about. There used to be thickets, fields of that, acres and acres of one particular genotype of it. And now they all look like that. And you can go for mile after mile in certain habitats, and it's just the bones of Staghorn coral, and it's depressing. So how do we go from that to that and that to that? What happened? It's a lot of things. There's some major stressors, things that are really big that happen, and everybody says, oh, wow, you get it. And then there's some minor ones. So one of the major ones, and this was a game changer for the entire Caribbean, was a disease that hit the Staghorn and Elkhorn coral, just those two species. And they called it white band disease. And it was a band of oh, what did I do? Yeah, I thought you said that was, must have, anyway. Okay, I pushed it too hard. That line right along the edge there is where the disease is moving, and it'll move out and kill that whole fragment that whole branch. It could do it in as little as a month. And so these huge stretches of coral died off in literally a year or two, and very little was left. It depends on where you are and where you were in the Caribbean, but estimates as high as 98% of them died, usually at least 90%. You know, you can talk about that all day long, but you get in the water and you realize, wow, it's really bad. Another thing was the loss of the long-spine sea urchins. Now, I used to hate those things because I was a fisherman and they were always stabbing me in the fingers and getting in my way, but they were really important. I use the analogy a lot of if you go to Disney World during the day, you know, there's all these people and they're doing all this stuff and bumping into everything and kids are spitting gum on the ground and all that. At night, when everybody goes home, these people pop out of the ground and out of little silent doors and they scrape the gum off the sidewalks. They paint every little thing that's scratched. I mean, they make Disney World look like new. And you come the next morning and it's like Disney World again and everything is lovely and great. And the urchins were like that too. At night, they came out and they grazed and they ate all the algae. When they died, it was like you fired the guys that cleaned at night and it just started to go downhill. So it's been bad. And we probably had too many sea urchins back in the day. There was probably too many of them, but they were an important component of the Caribbean reef ecosystem. The role of cleaning algae is usually taken up by sea urchins, crabs, you know, some of the invertebrates and fish, you know, parrotfish and tangs. And so the sea urchins mostly died off. There was like 98% of those died in the entire range of the urchin, all the way from South America to Bermuda. They all died in 6 months. So it was huge, it was devastating. It was a bacterial disease. It started in the Panama area. They suspect it might have come through the Panama Canal. And it was a bacterial disease that was transported by the water. And it was extremely virulent and it killed just like 99% of the diadema probably. So whatever's left might be resistant to it. The disease might still be there, but we might have a resistant population. But it's been real slow to recover. What I found out is that everything likes to eat diadema. You wouldn't think so, but it's like chocolate-covered peanuts, and everything gobbles them up, especially the babies. So it's been a slow recovery. Where you have a lot of fish, the recovery has been slow. Where you don't have any fish, like places in Haiti and Dominican Republic or Jamaica, they've recovered pretty quick. So another one is coral bleaching. You hear a lot about that. This coral here would normally be a brown color, but when the water temperature gets too hot for too long. Now, you can have a hot day, that doesn't do anything, but when you have hot day after hot day, they call it thermal heating weeks. So it's like 6 or 7 weeks above a certain temperature or a certain temperature, whatever, 2 temperatures there. These corals start to expel the zooxanthellae, the symbiotic algae that lives inside them, and they turn, instead of brown, they turn white. The algae that lives inside the coral is its main food supply. So when the algae is expelled from it, the coral starts to starve. It turns white like that, and a lot of times they die. So we've had several different years of that. The most recent was 2015, where it was a global bleaching event. You heard all about Australia, the Great Barrier Reef dying, yada, yada, yada. Well, it certainly happened here in the Caribbean. We've been having it for years, and it's killed a lot of corals. But there are corals that can handle the heat, and we'll get into that real quickly. And there's also hurricanes. Now, a hurricane's a natural phenomenon, but it really screws things up. And if you get hit by a hurricane, if you ask somebody that lives in Barbuda or Puerto Rico, they know all about hurricanes on land, but underwater, it does the same kind of damage. So moving on. So there's also these background stressors. These are the things that happen in the background. You don't really know, you don't see them. You don't think about sewage treatment. Most people flush the toilet and never think about it. But that stuff, if it's not properly treated, it leaches into the groundwater, leaches out onto the reef. It's nutrients. So there's this thing happening. These are just background stressors. It's like if you have a stressful job, you can do that for year after year, but it's grinding away. It's releasing hormones and enzymes and things into your body that are killing you. And that's the same thing that's happening. All these things, you have these nutrients from the farming, from stormwater runoff, development, overfishing, pollutants, African dust, that's a whole nother story. But you guys, if you live here year round, you know what African dust is all about. And there's pathogens that are transported from Africa to here that can kill coral. So it's a big deal. And then, of course, climate change and ocean acidification. it's becoming more of a foreground, but it's still kind of a background subtle stressor. So all of these things are stressing corals, some more than others, depends on where you're at, but depends on what's going on. So why am I wasting my time trying to grow coral back? You know, it just seems like, duh, this can't work. You're just stupid. And you know, climate change is going to kill everything. So why are you doing this? The water's so polluted, the corals will never live. Just give it up. It's all dead. You know, they're never going to live. What about ocean acidification? You know, and most people don't even know what that means or is, but it's this thing that the scientists say is a problem. It is. Or fill in the blank. You know, there's 1,000 reasons not to do something, but there's still hope. There's still things we can do. And so I like to say, you know, conditions in a lot of places are still favorable for corals to grow. There's still corals out there that can live in these and thrive in the present conditions. There's corals that are tolerant of the water conditions that we have today. And there's things we can do to change the outcome. If we do nothing, the outcome is pretty much certain that these coral reefs are going to continue to decline. There's too many things working against them, but there's things we can do to change it. So what can we do? We could develop awareness and hope. People need to understand why these corals have died, what the problems are, and that there's hope. I used to say my job is I'm selling hope, and selling hope and I'm buying time. If I can sell enough hope, if I can give people enough hope that they invest in this future, then we can buy some time. Can we buy enough time? I don't know. I wonder whether over the last few years, I'm wondering, can we buy enough time? But it's worth trying, and it's better than doing nothing and just hanging it up. So I'm doing that. We can eliminate some of the background stressors. So a lot of places have gotten to better wastewater treatments. I live in the Florida Keys. We've gone, the entire island chain now is on advanced wastewater, tertiary treated wastewater. So you could drink the water coming out of the sewage plant. We don't, but it's potable water, almost. But still, we need to be doing those kind of things when we can. And we did develop and implement a recovery plan. So that's kind of where we're at here, I think, is, you know, what can we do, and what would recovery look like? What would restoration look like here in Antigua? So to that effect, they've done a site assessment, Jen and Molly, and I came down and have done a quick little look around, and nothing like what they've done. That's step one. It's like, what do we have? What's left? What are the stressors? What are the problems? And how can we deal with it? And then develop realistic goals. I mean, we're not going to go out there and restore the whole reefs. What are we going to do? What reefs do we want to pick, and why? And what are the criteria? And is that a realistic? And I think it's real important to have a realistic goal for the different reefs that we pick to restore, and how long it's going to take and what can happen. And then we'll develop a nursery program where we grow the corals in nurseries, and then we'll start putting them out. So the nursery program will last a year, year and a half. The restoration program will be ongoing, and we'll monitor, we'll report. You monitor, you change things. So, well, that didn't work, you know, we'll try this next time. And luckily, for your benefit, a lot of these things have been tried over the last 20 years, and we've learned a lot, and you won't have to make all the mistakes I've made. We can make new mistakes, but we've learned a lot. So anyway, that's kind of the scenario for a recovery plan. And, wow, too fast. So I'm going to start with the bad news. So this is kind of what I saw while I was down here. And I don't want to dwell on the bad. I mean, it's bad. This is a thicket of elkhorn coral, and there's mile after mile of this, sometimes shallow, sometimes deep. Lots of coral has died. I mean, it's depressing. Martha saw this, and I think this is why she's so adamant about, you know, you get sad thinking about it, but you also like, what can we do to fix it? So this coral has probably only been dead, you know, maybe 5 or 10 years, but there's just, that stuff is everywhere. And, you know, I don't really want to, I see that, it just tells me, okay, corals used to be here. That's the site we can restore, you know, but it's not quite that easy. This is York Island, right behind York Island. And there's places there where it's just a complete dead zone. And I'm not sure why it all died, but it's dead. And yet not all of it's dead. There's still Some of the target species, some of the other species are still living there. So it's not completely dead. It's not a total waste, but that's the scene we see in a lot of these areas. So it's bad. And yet we're seeing a recovery of this diadema, these sea urchins, more on the sheltered areas, but out on the reef, out on 10 Pound Bay, there's a lot of these sea urchins. They hide again. They're down in the holes during the day, and at night they pop out and they clean. And you can see where they've been working, and you can see them that they're there. We're also getting larval recruits. So these corals are reproducing successfully, and you're getting larval coral settling and starting to grow in some areas, usually in the areas where the sea urchins have been grazing. So we saw a lot of these today out, I'm not sure where we were, but where? Falmouth? No, in the south. Okay. They just take me there, I'm sorry. I think, anyway. And a lot of those colonies have gotten big. They're a couple meters in diameter, and there's lots of them. There's also thickets of elkhorn coral still living, and you can go through thicket after thicket of dead stuff, and then all of a sudden you run into something like that, and you go, oh my gosh, this is awesome. And so one of the things that we would do in our nursery programs, we'll go back to that, we'll take a few cuttings from that one, because obviously that's a winner. It's not water quality, it's not something else, it's genetics that makes that coral live and thrive like it is. And then we found this thicket of, it's the fused staghorn, it's the hybrid between the staghorn and elkhorn, and it was huge. You could get inside there in that thicket and look around, and you couldn't see out of it without seeing extensive coral. So, there are some hope spots nearby that are really awesome. There's 5-star. I told Jen, I said, this is 5-star, this is awesome, I love it. And there's still shallow water habitats that have this type of coral growing. There's still staghorn coral, elkhorn coral in these shallow water habitats. So there's still places where this stuff is doing great, and there's hope. There's no reason why we can't, try to duplicate that in the right kind of habitats. So we're going to develop a nursery program. We'll probably use this tree idea. This is something that I invented in 2010, just out of desperation. When I started doing this back in the day, there was no textbook. There was no, we had the internet, but Google was a brand new word when I started doing this. So it was a while ago, and I had no resources to go to. So we invented this, and now this is probably the most common nursery structure throughout the world, all over the place. We were out today, and this guy had made one, It wasn't exactly like this, but the idea was there. So it's kind of exciting to see it. Elkhorn coral, so there's a lot of different corals we can grow on these trees. We've modified them to grow just about every species of coral out there, and it's been exciting to be involved in all of this kind of stuff. So just to give you an idea how fast these corals grow, you can take a pinky-sized coral, hang it on a tree over there on the left. That's March 14th, 2015. Here's basically a year later, the same tree. This is down in Mustique, down in the Eastern Caribbean. You know where that is, obviously. And that's the exact same tree with the exact same corals. And just to see what can happen in 1 year. These corals are kind of like growing weeds. They're easy, and so it's a good beginning coral, but it's also a very important structure building coral, and we're going to be certainly working with it. So as we start to develop the nursery phase, we're going to be looking for the corals that have survived all the different things that have happened. Anywhere from, we'll do them offshore, we'll do them near shore, shallow water, deep water, all these different habitats, we're going to be looking for them. And then we're going to look for the ones that have survived the white band disease, the bleaching events, and even the sargassum floats. We might find coral out in this bay that have survived the sargassum floats. And if you haven't been here in the summer, I would imagine this bay can fill up with sargassum. It starts to rot, starts to die. Oxygen levels go through the floor. But some corals can just hold their breath and get through it. And we're looking for those corals, and I'm sure we'll find some. So that's what we'll culture. That's what we'll try to plant on the nearshore reefs. And so there's hope on that. I was talking with somebody one of these nights. I was in Colombia, and we were doing some coral collecting down there. And the area we were at, they had diverted a river. I don't know, I think it was 1974. They diverted this main river near Cartagena. And now it came through a canal and just dumps mud out onto the reef. And we were there during a rainy season. and we were swimming around groping through the mud trying to find corals. And if you dove down below it to get to about 15 feet, it was clear, but up above it was really bad. We found this coral. The top of it was almost at the surface. It had water like hyacinths, freshwater plants stuck in the branches of it. And yet it was just the most fantastic coral, just as healthy and happy as can be. Every other coral in shallow water we found was dead. We found some deeper, but everything else was dead. But for some reason, this coral could just, like, I love the muck, you know. So we've gone and looked for those kind of corals throughout the Keys. Everywhere we go, we're looking for these in the most unusual and unexpected places. So another good example is, and I'll click through these quickly, this is fire coral, but the example here is really interesting. Let me back up just a couple. So sometimes this coral you'll get two different genetic strains that might settle in the same area almost side by side, and they'll both start to grow up. And in this case, right on this area right here is a seam where this particular genetic strain is meeting this one, and they don't fuse. They don't get along, but they're coexist. And so right along there, there's a seam, and you don't normally see the seams, but in an event like this where the one on the right is susceptible to bleaching, the one on the left is not. And it just, you know, becomes real obvious that some corals can handle the bleaching and some can't. And so when we get a bleaching event, a lot of times we're out looking for the corals that are still brown and happy, and we collect those. So it's a process. So once we start the nursery program, we're going to go out, we're going to look for corals, you know, these super corals, but also these corals that are living, you know, the thickets, the ones in weird conditions. And in this case, the example will all be elkhorn coral. We'll cut a little hand off. It might be the size of my hand. You know, we'll probably cut it right there. We might cut, you know, 5 or 6, maybe 10 cuttings off of that one, or maybe some of the ones around there. We'll take them back to the nursery. They might look like that. And then we'll split them into pieces. You'll use a pair of cutters. And this sounds brutal because most people say, oh, you can't touch coral, it'll kill it. Oh, you should see what we do to it. But it's really strong and really hardy. And so these corals, we cut them into little fragments. We then, so this is just that same coral. That's how many fragments we made out of a couple of these hands. And then we use monofilament, and we put monofilament loops around the coral. There's like a little, if you fish and make your own lures, there's these little crimps, little aluminum crimps. We use that to tighten up on the coral. And then we hang it on these branches just like that. And that's just kind of an example. We were doing some research on that one, so every coral is tagged. But those corals just kind of hang there on the branches. They're all cut up. They're injured, but they recover quickly. And you know, here's an example of how fast they grow. This was just down in Bonaire, April 2012. Just a few months later, the coral is already healed. It's already starting to grow some stuff out the side right here. And not even a year later, you're getting like a hand of coral coming out of the bottom of that. And so what we do is you might have a tree like that just full of these corals. We're going to get in there and we're going to cut all these hands off. The hands would look like that. They're the size of your hand, sometimes a little bigger, sometimes a little smaller. So with the hand, we can either set up another tree, cut them into more fragments, just like we did in the beginning, put them back on a tree, or we can take them and put them out on the reef. And I'll go through that process real quickly too. So again, we've developed, for the restoration phase of it, we want to have realistic goals. You know, we're not just going to go willy-nilly planting corals everywhere. We're going to say, okay, this is an area where the water quality is good. It used to have this particular coral. Every indication is that we could put it back and it would thrive. You know, why am I saying that? Well, I've done it enough times to know that you can kind of eyeball it and you know what's going on. We'd start out planting, we'd monitor, make some adjustments. If something's working, great. Continue doing that. If you need to adjust, if something's dying, then you make an adjustment. You change it around, you keep going. But always monitoring the progress, you know, annual reports, whatever. That'll be Molly and Jen doing that. But I think they like doing that. Anyway, so this is just an idea. These are so if the coral didn't go back to the nursery, we would plant these little things on the ground. We'd use an underwater epoxy to glue them down. They would turn into clusters like that in less than a year or two. They would start to look like that. These are, this is down in Mustique. These are about 3-year-old corals. They started the size of my hand. They're now each one, you know, half a meter to a meter in diameter. And in a very short period of time, you can start to restore a reef. And there's just some of the images from down in Mustique. So for Staghorn coral, we used to do the same thing. We'd glue every coral down. We're changing our ways. One of the things we've done in several places in the Caribbean now is we set up a bamboo frame on the bottom. So bamboo is eventually going to rot and fall away, but it'll stay for a year or two. So we cable tie corals onto the bamboo frames. This is one that's about 6 months old. In about 12 months, that's what they start to look like. These things start to grow out. They wrap around the frame and they start to expand. That's 24 months of, you know, growth of this particular coral. And that's what a thicket looks like that we've created. We've created dozens of them in Bonaire, Curacao, and Colombia that are sometimes, you know, the size of this entire, not just this room, but, you know, the area out there and over there in just a few years. So it doesn't work like that everywhere, and it doesn't work like that with every genotype, but it can work like that. And so it's pretty exciting, and you don't see the frames anymore. It's just an intertwined thicket of coral. So exciting stuff. So kind of conclusion, and you know, I'm okay on time, but not bad. So there's hope. It's bad, but there's hope. There's still some really great spots out there. We've spent a lot of time now, 20 years, figuring out how to do this and do it on a big scale. And I'm not going into it on this particular talk, but I started a lot of this stuff, and a lot of people are following me and doing what we started. And I call that Restoration 1.0, but I'm on to Restoration 2.0. And 2.0 is coming fast, and it involves not just 3 species of coral, but 25 different species of coral. And for these corals, it's a whole new game, and it's a order of magnitude more efficient and faster, better, cheaper. It's really exciting. So, but if you look at that, this is a restored thicket in Belize that a friend of mine's working on, and it's full of little fish now. And that's the goal, not just to make it look pretty, but attract fish and make it, you know, make the reef come back to life. So that can be done, and measurable improvements can be made. You know, we're not going to restore every reef out there. We're going to pick a few reefs and a few sites that we think people would enjoy or that are strategic, and we're going to restore those. And then we'll see where it goes, you know. You just, I don't know how big your vision is. My vision is big. My vision is island-wide, but you start small and you prove yourself. So I think that's my last slide. So this is, again, a restored area in Belize. There was nothing here about 6 or 7 years ago. It looks like that now, and you can't tell the difference between a restored area and what it used to look like. So it's super exciting, and a lot has happened in the last 10 years. We learned a lot in the first 10 years that I was doing this, and the last 10 years has been, you know, kind of the gravy. We're finally seeing results. We're working with a lot of really exciting corals, and it's starting to become even more fun than it was 20 years ago. So that's the end, and I don't have anything else. Is there any questions?
Ken, I know the sea urchins help clean the place up. Are there other species that also do that we could identify?
Yeah, so that's a great question. So the sea urchins are one of many different critters that play an important role in maintaining the reefs. And the diadem of those sea urchins were the most, there's other sea urchins out there that didn't die, but they're not as important. So parrotfish, I know everybody likes to eat parrotfish here, but they're really important. The tangs, and a lot of places eat tangs too, they're important. There's several different species of crab that pick and eat algae. So it's an ecosystem, and when you disrupt it by fishing parts of it or something dies like the sea urchins, it just it causes a lot of problems, unintended consequences. And you know, the parrotfish is a big issue, but I think the sea urchins really is a bigger issue. If you could get the sea urchins back, they just have such an important role. And then everybody always asks, well, aren't you culturing those? Those are the hardest little boogers to culture that we've ever worked with. There's a friend of mine, he's one of the best aquaculturists in the world. He worked on it for 15 years, and he's like 80 years old now. He said, I'm handing it over to somebody else. We're making progress, but it's difficult. They are so hard. to culture. I think there's a future in that, but we're not there yet. So we're just waiting for a natural recovery.
Ken, thanks for a great presentation and for giving us hope.
Yeah, that's what I wanted to do.
Question is,
selling hope.
Can you speak a little bit about how you work with the local communities in all of these locations, the local fishing community, people who live along the coast, to help mitigate or actually work with you on some of these projects, so they're helping as well?
Well, so we always believe in local ownership of the project. And we've worked with a lot of these different, everywhere we go, really, we've reached out to the local community. Now, sometimes it's more challenging than others. A lot of places it's spear fishermen, and they don't want to listen to us. I'm not worried about the spear fishermen. I'm worried about the trap fishermen and the net fishermen. But we've done that. It's a local project, and so a lot of that falls on the local people. I mean, we've certainly emphasized the importance of engaging the local community, especially the fishermen. But as far as me actually making all those connections and making it happen, I can't. I'm not the local guy. But I certainly think that's a super important part of the success of any program, because these people need to see that you're not there trying to put them out of business. The last slide, well, not that slide, but look, we're building habitat for fish. Fisheries are an important part of. We all like to eat fish, and we want you to keep fishing. We want you to do it sustainably, and we're trying to put the coral back so that there's more fish. It's a complex thing. In some places, I was a fisherman for 25 years, so I know what I've been to the meetings, but I know how important it is to bring them alongside. And if you can get one or two important fishermen on your side and you win them over, then all of a sudden, all the younger fishermen and the up and coming ones, they start to pay attention. So if you can find that a couple of key people, that's you're on the right track. I hope that answers your question. Yes, sir, back there.
Great presentation, Ken. Thanks very much. I was just surprised. So much of it seemed like it wasn't us. It sounded like there was disease, there was temperature. Have you mapped sort of population correlation with dead coral? I mean, is it to do with too many people, or is it really actually more to do with climate change, which of course itself has to do with people? But
well, I mean, it's a lot of things, and the core of it, a lot of our problems, if we hadn't had a disease that wiped the elkhorn and staghorn coral out in the 80s and the loss of these sea urchins, things would look different. I've done a lot of diving, and I've been to Jamaica. So Jamaica has not done anything to fix any of their problems. They still have the same runoff. They have more people now than they did 10 years ago. Nothing has changed, but there's some reefs where the sea urchins have come back. And you could literally go from an area that's covered with sea urchins, the reef looks fabulous, and in a 10 or 15 foot space, you'll transition from lots of sea urchins, beautiful reef, not a lot of sea urchins, marginal reef, no sea urchins, and trash reef. So, there definitely people have an impact. There's no question about that. We're screwing it all up. We're fishing it. We're polluting it, all that. But it's a fairly resilient system when a lot of the pieces are in place. But when you pull these important pieces out, then it collapses and it's not going to recover until some of those pieces are put back in. We're trying to put the coral piece back in. The sea urchin piece, we've been trying. We haven't gotten anywhere on it. It's a complex. I can't say that any one thing, it's this or that. It's a whole bunch of things, and every area has a different suite of a mix of those things. But yeah, we're the problem, but we can be the solution. So yeah, what makes a coral grow on the tree? I don't know. We started hanging them like that, and who would have known? So we started growing them on the ground like a typical coral should grow. And then we saw some people working in Jamaica, and they were growing them on lines. And we said, well, let's try that. And we found out they grow 2 or 3 times faster hanging from one of those trees or from a line than they do on the ground. And I have not gotten a good answer for why they grow faster, but they grow really fast. It's really easy to take care of them. And it's changed the nature of restoration. We can now grow way more corals than we can plant. So the goal now is to plant more. That's exciting. Did you have a question back there? Well, you know, corals will eat plankton. At night, the polyps, so each of these, I can't explain it. It's a colonial animal, so there's hundreds, thousands, millions of little polyps. Each polyp is kind of its own mouth and its own stomach and its own digestive system, but they're all networked together. And at night, the polyps extend tentacles out, and so the tentacles can grab and catch plankton in the water. But their main source of food is this algae that lives inside them that gives it, all these corals that are brown, there's an algae living inside them. And during the day, the algae produces food for the coral eats it, and it's a happy relationship because the algae can't live without a host. So that's its actual main source, and that's why when they bleach, they're actually on a diet. They're starving to death because they've gotten really bad at catching plankton. Unless they're a deep water coral, they've gotten kind of lazy about plankton. You could actually grow them in an aquarium and never feed them. And I mean, they wouldn't grow real fast, but if you give them the right amount of light, they'll grow. So.
Hi, when a replanting is unsuccessful, why is it usually that it doesn't take? And have you figured out a way to resolve whatever impediments you come across?
Sometimes I think you run in, you plant them in an area and the water quality is just not that great. It might seem okay on day one, but then it's like if we planted in the bay right now, yeah, the bay looks pretty good. The water's clear. Hey, let's do it here. This is great. And then the sargassum mats come in and cover the bay. Oxygen levels fall to zero and everything dies. And so sometimes there's unanticipated problems. Sometimes there's predators. We planted them in one area where, and we didn't realize this, but at night, these worms would just crawl out of the rubble and out of the reef and just eat the coral like crazy. And we finally was like, what's going on with the coral? Why are they all dying? We went out there at night and there was these fireworms all over them, just eating them up. So, you know, you kind of learn, I mean, we've learned a lot. You're still gonna make mistakes, but sometimes, you know, you look at, you know, this coral used to live here. Maybe there's still some there. So you get better at guessing on where to put it. And then sometimes you just say, we screwed up. Let's not plant any more there. Move on. Yes, ma'am.
Yeah. You go and harvest a hand, you cut it into pieces, you put it on a string, hang it from a tree. How quickly must you do this? How long can the corals survive out of water?
So we can do that underwater or we can do it on a boat. So we did a test to find out how long you could keep a coral out of the water. And surprisingly, a long time. We did a test with the Staghorn coral here, and we could keep them out of the water for up to 2 hours with no mortality, out in the sun with no air, you know, just kind of out. And yet they ship corals dry with just bubble wrap. They'll ship them, you know, we could ship them FedEx to, you know, Washington State overnight and they do fine. So they're actually pretty tough. But yeah, a lot of times we do it underwater because I want to reduce any stress on the corals.
Hours. I mean,
oh yeah,
if you're on the boat.
And we try to engage more volunteers by doing it on the boat or on shore, and they get to see it and be a part of it. And you can do, you know, you can handle a coral and do all the processing in 20 minutes. Yes, sir.
Curious, having swum Exchange Bay for over 62 years.
Oh my gosh, I'd like to talk to you.
And seeing the change in the coral. But one thing, you had one slide that had brain coral. Is there any hope for the brain coral?
Well, we're culturing and growing brain coral in Florida. The bad news is, and I don't want to depress you any, but there's a disease right now that it kind of started in Florida and it's worked its way through the Florida Keys and now it's working its way through the Caribbean. And brain corals are one of the corals that it kills. And when it gets here, it's not going to be pretty. It's going to kill 80% of your corals or more. And I don't want to depress you. It's depressing. But we're, again, in the Keys, we're going behind the disease front. We're picking up the ones that have survived and we're culturing those and we're going to replant with the disease-resistant ones. But that's a needle in a haystack. That's a drop in the bucket of what needs to be done. That's anyway, yeah, brain corals are cool. I love working with them, but they're hard to Yes, sir.
So what's your actual prognosis on our reefs, and where do you think you could actually start to work on cultivating new reef?
Well, I mean, they're bad, but I didn't really see much active disease. And there's these hope spots that I mentioned, and even just in front of York Island and out here, there's still the targeted corals. They're not going to recover on their own. The reproductive success of these corals is down to almost zero. I mean, they struggle greatly to reproduce on their own. So if we don't do anything, it's not going to get any better. It's only going to get worse. I think there's places that we could certainly make headway. I mean, we might have setbacks, but I think we'll learn along the way. I think there's certainly the Florida Keys are much worse than here, and we're having great success there. So I think there's hope. You know, let's be realistic about what we can do, but let's try something. And I think you'll be real pleased with what happens. You know, they grow fast. Yes, ma'am.
Thank you so much. This really gave us a great education. You talked about a wide range of geographies, and I wondered about how specific the ranges of the corals are to those locations. So can you bring coral from Mustique to Antigua, and is it all just similar Staghorn and Elkhorn, or is there a more narrow geography where you need to replant them?
Well, we tend to, when we set up a project, we tend to use the local corals because the local corals are the most adapted to that area. But what we're starting to see throughout the Caribbean is that there's genetic bottlenecks in different areas. When we go to, out here, this particular staghorn coral is all just kind of weedy. You know, it might be the size of a cigar, you know, your thumb. It's just kind of weedy coral. We go to Bonaire, the stuff is the size of your arm. It's just huge. And so genetically, they still have a better, a richer genetic population. So the future will probably involve not bringing corals per se from Bonaire or somewhere else, but bringing the gametes there. They can cryopreserve the sperm from corals quickly and readily. And so that'll probably be the first thing that gets transported. As a matter of fact, we've done that with elkhorn coral from Curacao to Florida. And in the next year or two, they're going to be bringing Bahamas corals to Florida and Florida corals to the Bahamas, the gametes, and doing that. So that's kind of the future. Right now, they're too nervous about, well, if you brought a coral from Bonaire to here, what else is on that coral? What, you know, it's just like too many unknowns. Let's start small. But I think that's the future. I really do. That's a great question.
We have a question from our members on Zoom.
Okay.
Allow us to read one question first from our members on Zoom.
All right.
Okay, the first is, what happened to all the fish that used to occupy the reefs when living?
What happened to the fish? Well, they probably got eaten. Yeah, that's a good question. They are displaced. The fish will attract, they'll aggregate around a dead, I don't know who I'm looking at. Where's the camera? They'll aggregate around a dead structure. You can put a shipwreck out there and fish are going to aggregate around it. But when a reef dies, what happens first is I think recruitment stops. The larval fish are really attracted to the smell and the sound of a live coral reef. And so they're drifting by and they don't smell it anymore, so they don't drop out of the plankton, and you just don't have a new fish coming. But yeah, what happens to them? They probably get eaten. I mean, fish don't live forever either. So, but as the habitat dissolves, the fish are habitatless and they get eaten. So that's the best answer I can give. I think they had another.
Yes, we have 3 more on Zoom. The second is,
Sorry.
speaking of Mustique, today there is volcanic activity nearby St. Vincent. How does volcanic ash and other outputs of volcanism affect nearby reefs?
If it's too close, it's going to kill it. Our nursery will actually probably be fine because the corals are hanging up in the water column, and any ash that would settle on it will just fall off. But the reefs themselves will be impacted. If they have a lot of sea urchins, the sea urchins will try to clean it up. Wave action might move it around. If it's a lot of ash, yeah, they're toast. They're going to die. But yeah, hopefully, hopefully it won't blow to Mustique. And one more.
The next question is, isn't this approach a bit Darwinian? Perhaps we should look forward to the future rather than preserve the past?
I would like to have coral reefs for my grandkids to see, and so no, I'm not about to just roll over and die. You know, it's easy to give up and quit, but we've brought this on as a human species. We've changed the climate and we've changed the reefs. And I think it's incumbent upon us to try to do what we can. You know, national parks and other restoration efforts have been successful and have, you know, maintained something that we all treasure. And I think people treasure coral reefs. And I think it's worth fighting for them. And that's, you know, I could disagree on that one.
We have time for one more question, Ken. One more question.
Oh, there was a question up here too.
Wonderful presentation. This is really a related question. How would you encourage people to think about the costs of letting the reefs go? In other words, if you don't do this.
Yeah, I mean, I think it's immense. They call it ecosystem services, but, you know, tourism, there's fisheries. The cost of not doing something is just mind-boggling. And so the cost of trying to do something is, you know, it can be expensive to really do it right. I mean, we should be working on, you know, climate issues. There's a lot of things we can and should be working on, but some of them are so big, it's so easy to say, oh, you know, what can I do? Well, this is something we can do. So I'm like I said, I'm trying to buy time. Can I buy enough time? I don't know. So I've got a plan B and a plan C that I'm working on, trying to drum up some interest in. And it's a doomsday thing. And I don't really want to talk about that much, but I think there's, you know, we have to start thinking about what if we can't do this? You know, what kind of plans can we make? You know, there's gene banks or seed banks all over the world, and I think we're going to need to develop a coral seed bank. But I don't want to dwell on that. I want to dwell on what can I do in my lifetime, and can we bring this back? So I think
yeah. So we had a lecture last week, Walter Isaacson, on breaking the code genetic editing. We're talking a lot about, you know, is there any work being done in that area?
On the genetic?
Yeah, if there any intervention that can be made to create stronger species?
Yeah, there's actually a lot of work going on, especially in Australia, but other places. They call it assisted evolution. So right now, you know, nobody's actually trying, well, they might be trying to, you know, edit the genes of these corals. We're just to the point where we're understanding which genes respond to heat, which ones turn on, turn off, and heat, cold, other stressors. So we're just now really starting to understand that. We've just gotten to the point where we can sexually reproduce corals, raise the larvae, and put them out on the reef. So a lot of that has just happened in the last, you know, 10, maybe 15 years, and we've started to get really good at it. So the idea is probably at first is the selective breeding idea, this idea of bringing these strong genetic strains from somewhere else, breeding them with your local population and trying to do that. And then as you're doing that, you're also saying why is this coral successful? What genes are making it successful? And then in the offspring, not all the offspring will have the genes that you want. So which ones do and which ones don't? And then, you know, maybe it gets to gene editing. I don't know. It's a really big issue when you swim out there. You know, just Antigua, you know, we swam mile after mile of dead coral, and so bringing it back on a scale that it's like is mind-boggling. You know, we can do this in a lab, but how do you turn that into something real? But it's a super exciting time in coral reef biology and all that stuff. And I mean, it's running past me and my ability to understand it. I'd rather get out there and just, you know, do my thing. But it's really exciting, and I don't know where it's going to go. But the clock is running fast towards local extinction in a lot of areas, and you know, I think we can intervene and slow that down, and maybe slow it down enough that, you know, we as a society can get our act together and not continue to treat the earth like a toilet. So anyway.
Well, ladies and gentlemen, thank you all for coming. And Ken, thank you so much for a great presentation and also for all your terrific work this past week. And we cannot thank you enough for everything you've done to get us moving in the right direction. So everybody, if we could just say thank you.
My job is to help you, and that's what I'm going to try to do. And you know, I appreciate people like Arthur and the whole gang here that's, you know, trying to get behind this and make a difference. And I'm, you know, you got a couple of great gals here. Get behind them.
Thank you, everybody. Have a great evening. It's good. Ken, super good.